All right. I think we'll get going. People are still joining, but I think maybe we'll get started slowly and everybody will start to come in. We're approaching 60 participants right now. Welcome everybody to a sort of special edition of the CRCHUM Seminar Series. Bienvenue tout le monde. C'est vraiment un plaisir de vous présenter aujourd'hui notre conférencière locale, mais plus prestigieuse quand même. We have a wonderful lineup for you today that is really focusing on sharing knowledge that exists within the CRCHUM community and helping each and every one of you in various animal modeling endeavors that you might be involved in as part of your projects. So this atelier or workshop is really to help provide critical information on how to improve performance of the models you're working with, as well as offer some very practical advice for the development, the maintenance, the breeding, and use of those animal models. So today we have 4 speakers lined up for you today. Christian Demers, who is, of course, the chair of CIPA and has had more than 10 years experience at the CHUM, I think, because he was here when I started, and has been a significant resource to me over the years. We have also Dr. Hélène Héon, who is, of course, the head veterinarian here at the CRCHUM, and again, an immense resource to my animal modeling experiments over the years. We also have Dr. Jean-François Schmouth, who is a Responsable technique for the platform for the modélisation d'animaux and transgéniques. I always forget the official name. So JF will share with us his insights on developing new models, especially with the new technology of CRISPR. And finally, we have Dr. Thierry Alquier, who is head of the Cardiométabolique, but also a researcher that uses Krelox models extensively. And I'll wrap up the afternoon with sort of some take-home messages to consider. We will take questions at the end of the 4 presentations, as we suspect there'll be quite a bit of overlap and maybe things will become clear with each subsequent presentation. So without further ado, c'est vraiment un plaisir de vous présenter Christian. Christian, take it away. Merci.
Merci beaucoup, Christine.
You have.
Oops.
Oh.
OK, j'ai un petit problème de...
Problème de remote control? Attends une minute. Do you have control yet?
No.
No. We did test this.
Yeah, I know.
No? Okay, I can advance the slides for you, no problem.
Okay. Diapo suivante, s'il te plaît. OK. Lorsque Thierry m'a demandé de participer à cet atelier, en fait, j'étais un peu surpris. Généralement, la partie CIPA est laissée souvent à la discrétion des chercheurs et des utilisateurs du service de l'animalerie. Mais je suis assez content qu'il ait pensé à moi pour cette conférence-là. En fait, je veux un peu vous donner l'ABC de ce qui doit être fait avant de partir sur le chemin de la création d'une nouvelle lignée de souris. Donc, le titre un peu illustre ça. Là. Donc, du point de vue du CIPA, là, ce qui doit être fait avant de partir le processus de création des souris. Donc, diapo suivante, s'il te plaît. OK, donc, tout comité de protection des animaux, en fait, un des buts de ces comités-là est l'application du principe des 3 R, donc remplacement, réduction et raffinement. Il faut bien comprendre que lorsqu'on veut créer une nouvelle souris, il faut se poser quelques questions préalables. Donc, la première, bien entendu, est-ce qu'on peut répondre à nos hypothèses de recherche sans utiliser d'animaux. Donc, on peut probablement déjà dire que si vous pensez à une nouvelle souris, vous avez déjà réalisé que ça vous prenait absolument un animal. Mais depuis quelques années, il y a quelques ressources qui sont disponibles, comme entre autres Society for Human Science ou l'Université de Windsor, qui a un site assez complet sur les alternatives sur l'utilisation des animaux. Donc, dans un contexte de recherche, je pense que c'est important de répondre à ces questions-là au préalable. Il y a tout dans la même optique, si un animal est nécessaire, est-ce qu'il est vraiment nécessaire de créer une nouvelle lignée de souris? Donc, c'est également une question qu'il faut se poser lorsqu'il est question d'une nouvelle souris. Là. Je ne sais pas si vous êtes au courant, mais si, par exemple, vous allez sur le site de Jackson, juste pour les modèles de recherche pour le diabète, je ne me rappelle plus le nombre de pages, mais la dernière fois que j'étais allé voir, là, il y avait plus de 100 pages de modèles génétiques pour le diabète. Donc, avant de créer une nouvelle souris, ça vaut peut-être la peine de faire quelques recherches. Diapo suivante, s'il vous plaît. Okay. Le deuxième principe, comme je disais tantôt, donc la réduction. Là, ce qu'il faut bien comprendre lorsqu'on parle de nouvelles souris, c'est que le type de souris qu'on va créer va avoir un impact sur le nombre d'animaux qu'on va produire. Donc, si vous créez une souris, un simple knockout, vous allez avoir besoin d'un certain nombre d'animaux, d'un certain nombre de contrôles. Si vous créez des modèles un peu plus complexes comme des souris LOXP, des souris Cree, des souris Cree inductibles, là, à ce moment-là, le nombre de souris nécessaires pour réaliser votre projet de recherche va augmenter de manière significative, ne serait-ce qu'à cause des contrôles nécessaires dans vos projets. Donc, dans l'optique où vous voulez créer une nouvelle souris, toutes ces questions-là doivent être abordées là, au préalable parce que ça va avoir un impact important sur d'une part, sur votre budget de création de souris, là, puis à long terme, là, en termes d'espace pour héberger tous ces animaux-là. Donc, c'est vraiment important d'y penser au préalable. Diapo suivante. Finalement, le type de souris que vous allez créer va avoir également un impact sur les conditions d'hébergement. Pas toujours, mais parfois, les souris qui vont naître, bon, pour toutes sortes de raisons, ils ont besoin de soins particuliers dès le début de leur vie. Souvent, on peut observer qu'il y a des modèles d'animaux que les homozygotes ne sont pas... leur espérance de vie chez l'homozygote est plus basse que des souris normales. Alors là, il faut considérer qu'on va travailler soit avec des animaux hétérozygotes, soit on va travailler avec des animaux à un âge bien précis, ou même dans certains cas, ça arrive qu'on travaille avec des animaux juste avec des animaux durant la période fœtale. Donc, on n'a jamais d'animaux vivants qui naissent. Donc, il faut aussi considérer ça. Les ressources qu'on a au CRCHUM, Hélène, Jean-François, ça peut être des ressources importantes là, pour un ou l'autre des points de raffinement. Donc, aussi bien les conditions d'hébergement que pour établir le modèle. Diapo suivante, s'il vous plaît. OK. Si les 3 R ont été répondus, là, on arrive à l'étape avant de créer la souris. Donc, la première chose, en fait, qu'il faut se poser comme question, est-ce que la souris existe déjà? Donc, au CRCHUM, il y a au moins 2 ressources là, que vous pouvez utiliser. Il y a à la fois la plateforme dirigée par Jean-François ou encore le comité de protection des animaux. Donc, nous, on a accès, en fait, à l'ensemble des protocoles d'utilisation d'animaux du CRCHUM. Dans certains cas, vous pouvez nous contacter, on va vous dire si les lignées existent. L'autre ressource aussi qui est très importante, c'est qu'au Canada, il y a un réseau de coordonnateurs de comités de protection des animaux. Donc, pour l'ensemble des grandes institutions de recherche au Canada, on peut poser des questions à l'ensemble du réseau. Donc, c'est très possible de savoir si une souris est disponible au Canada. Il y a bien entendu les fournisseurs commerciaux là, de souris, là, comme Jackson, Taconic ou Charles River, qui peuvent être aussi des ressources. Donc, ça vaut la peine de consulter les sites web de ces compagnies-là. Il y a également d'autres ressources comme le International Mouse Strain Resources ou le Canadian Mouse Mutant Repository, qui sont aussi des bonnes ressources. Et il y a également le Mouse Genome Informatique, qui peuvent être des ressources intéressantes pour trouver des souris. Souvent, ces sites-là fonctionnent soit avec le nom du gène. En tout cas, c'est relativement facile de les utiliser, mais d'emblée, ce que je pourrais vous dire, c'est demander au comité ou demander à la plateforme dans un premier temps. Là, je pense que c'est un bon réflexe là, avant de commencer la création d'une souris. Diapo suivante, s'il vous plaît. OK. Quand vous êtes arrivé à l'étape du protocole, parce que, bien entendu, la plupart des comités de protection des animaux à travers le Canada, en fait, demandent à ce que qu'il y ait un protocole de soumis avant même la création de la souris. Donc, et bien entendu, il y a 3 choses que le comité va vous demander. Donc, ça vaut la peine de faire l'exercice au préalable. Donc, le comité doit comprendre c'est quoi les bénéfices pour la science, pour la santé humaine ou la santé animale. Puis, c'est ça. Donc, cette première justification-là là, doit être faite. Les 3 étapes que je vous ai expliquées sur les 3 R, mais il faut les mettre dans le protocole également parce que c'est vraiment important d'expliquer tout ce que vous avez fait au préalable pour appliquer le principe des 3 R. Puis finalement, le comité va également vous demander tout ce que vous avez fait là, pour trouver l'animal avant d'arriver à la conclusion que vous deviez en créer une nouvelle. Donc, pour tous les protocoles de création d'animaux, c'est vraiment la base que les comités vont vous demander. Puis ça, c'est peu importe l'institution où vous êtes. Diapo suivante. Une fois que la souris a été créée, donc là, ce qui est important, c'est qu'il faut que ça soit aussi expliqué dans votre protocole. Donc, il faut bien souvent, pour des nouveaux modèles, faire le phénotypage de ces animaux-là. Donc, c'est vraiment important que votre protocole décrive également votre protocole de phénotypage afin de prouver finalement que le modèle que vous avez obtenu correspond à ce que vous attendiez d'avoir. Donc, je sais pas, par exemple, si c'est un gène qui est une mutation dans un gène chez l'humain, puis vous créez la même mutation chez la souris, est-ce que vous obtenez le même phénotype que chez l'humain ou non? Ben, puis comment que ça serait quoi les observations pour répondre à cette question-là? Une fois que la souris est créée aussi, peut-être que le phénotype diffère un peu de ce que vous attendiez. Donc, il faut peut-être ajuster les conditions d'hébergement, aussi les conditions d'eau ou de nourriture. Dans une ancienne vie, je travaillais dans le laboratoire où je travaillais, on avait un modèle de souris qui devait avoir une diète spéciale dès la naissance pour survivre. Donc, c'était vraiment important que les conditions de nourriture soient particulières pour notre colonie. Depuis quelques mois, en fait, il y a également quelque chose qui est en train de se mettre en place au Canada. Donc, c'est l'évaluation du bien-être. Donc, dans le cas des animaux créés par génie génétique, c'est vraiment important que cette évaluation soit faite dès la naissance des animaux parce que ça va avoir un impact sur toute la vie de l'animal. Donc, déjà, à la plateforme que Jean-François dirige, il y a quand même une évaluation qui est faite dès la naissance des animaux, à savoir est-ce que la taille des animaux, est-ce qu'ils se nourrissent correctement. Il y a quelques notions qui sont faites d'emblée, mais toutes ces observations-là vont permettre l'établissement de ce qu'on appelle le passeport souris ou passeport rat, si on parle d'un rat modifié par génétique. Et ce document-là, finalement, c'est comme le document d'identification de l'animal. Donc, c'est vraiment important pour votre équipe. Donc, c'est une information là, qui peut suivre l'animal tant qu'il va être hébergé à l'animalerie, mais également c'est un document qui est vraiment important pour l'équipe de l'animalerie qui va s'occuper de vos animaux. Et aussi, ça devient un document d'importance dans le cas où vous décidez de partager cet animal-là avec une autre institution de recherche. Donc, toutes les informations concernant les critères le background génétique de la souris, les critères de repro, le type de repro qu'il faut faire, est-ce que c'est homozygote, hétéro, entre autres. Toutes ces informations-là doivent figurer au passeport souris. C'est vraiment une mine d'or pour tout le monde, là. Puis, au fil du temps, ce document-là peut être revisé à intervalles réguliers là, pour tenir compte là, de la connaissance qui est obtenue sur la nouvelle lignée. Diapo suivante. OK. Finalement, tout ce que je viens de vous dire depuis quelques minutes, là, en fait, ce qui était important aussi, c'est de noter le plus grand nombre de choses par rapport à votre modèle animal, parce que lors des publications que vous allez faire avec ces nouveaux modèles-là, l'idée, c'est de pouvoir rapporter le maximum d'informations de manière à ce que ces informations-là puissent permettre à d'autres équipes, finalement, de reproduire les expériences, le cas échéant. C'est vraiment une préoccupation depuis quelques années, la reproductibilité des résultats. Donc, on est capable on contrôle un certain nombre de variables en recherche. L'idée, c'est qu'à partir du moment qu'on contrôle ces variables-là, il faut qu'on puisse les reproduire. Pour que les autres équipes, les autres laboratoires puissent les reproduire, il faut qu'il y ait connaissance de ces variables-là qui ont été contrôlées. Donc, je vous ai énuméré là, 6 ou 7 caractéristiques de ce qui devrait être rapporté dans un article scientifique là, qui a été créé par le groupe Arrive. Donc, c'est vraiment important que ce genre de détails-là figure dans les matériels et méthodes des articles scientifiques. C'est vraiment l'ABC pour être sûr de pouvoir reproduire des résultats d'un groupe à l'autre. Merci de votre attention.
Merci, Christian. Merci beaucoup. La prochaine partie, c'est de Dr. Héon. Dr. Héon, est-ce que tu veux le remote control?
Oui, s'il vous plaît.
Parfait.
Est-ce que ça fonctionne?
It says it's waiting for you to accept.
Je l'ai accepté.
Parfait.
Ça ne fonctionne pas.
Non, il y a un délai encore.
Oui. Est-ce que tu veux changer mes diapositives?
Je peux le faire, oui.
Okay,
bien
merci.
sûr.
Alors, bonjour à tous et moi aussi je voudrais remercier Dr. Alquier de m'avoir invité à participer à cet atelier pour que je puisse vous parler d'une de mes champs d'intérêt, des bonnes pratiques d'élevage. Prochaine diapo, s'il vous plaît. Alors, la plupart de nos souris transgéniques ont été créées sur un fond génétique consanguin. Donc, les animaux sont isogéniques, tous les individus ont le même génotype et ils sont homozygotes à tous les locus. Cela, ça va vous permettre d'avoir des animaux qui permettent une bonne reproductibilité expérimentale pour les traits qui sont influencés par la génétique, bien sûr, et un modèle animal qui va être cohérent et uniforme pour les études. Une lignée consanguine va se transformer en sous-lignée après 3 générations de croisements non frères et sœurs et après 10 générations de croisements frères et sœurs. Donc, il est préférable de faire des croisements frères-sœurs. Prochaine diapo. On a 2 ennemis quand on fait de l'élevage consanguin. On a la dérive génétique et la contamination génétique. La dérive génétique, c'est la tendance qu'ont les gènes à constamment évoluer, même en l'absence de force reproductive. C'est créé par des mutations qui vont apparaître de façon aléatoire. Elles vont apparaître ou même disparaître et elles vont se fixer dans la colonie et cela peut affecter le phénotype de vos souris. Les petites colonies sont plus affectées que les grandes colonies, comme le montre l'image à côté. La dérive génétique, c'est inexorable. On ne peut pas la prévenir, mais on peut la limiter en utilisant des croisements frères et sœurs, en rafraîchissant la lignée périodiquement et en cryopréservant des embryons ou du sperme. Prochaine diapo. Donc, comment on peut limiter la dérive génétique en faisant un rétrocroisement de la lignée pour la rafraîchir? On recommande de faire un rétrocroisement après 5 générations. À ce moment-là, on va faire 2 rétrocroisements pour rafraîchir les chromosomes X, Y et le génome mitochondrial. Si on attend 10 générations, ce qui n'est pas nécessairement une erreur, mais à ce moment-là, on recommande de faire 3 rétrocroisements. Une fois les 3 rétrocroisements complétés, on établit une colonie dite rafraîchie et on utilise les animaux issus de cette colonie pour remplacer les reproducteurs qu'on retire de notre colonie actuelle. Prochaine diapo. Notre autre ennemi est la contamination génétique. Alors, c'est lorsqu'on croise accidentellement des souris de 2 lignées différentes. Comme la plupart de nos souris sont noires et elles sont tous hébergées ensemble dans la même salle, c'est relativement facile de se tromper de cache et de faire un mauvais croisement. On doit aussi considérer que si on a des souris de la même souche, mais de 2 fournisseurs différents, on peut considérer ça comme une contamination génétique si on les croise. Ça, c'est causé parce qu'il y a beaucoup de polymorphisme au niveau particulièrement de la souris C57BL6 et ça peut affecter le phénotype. Dr. Alquier va vous en parler un peu plus tard. Ce qu'on recommande, comme on ne peut pas séparer nos souris par couleur, on n'a pas assez des grandes animaleries, on peut tenter de séparer les lignées, au moins les lignées qui ont une nomenclature similaire. Il faut croiser, il faut génotyper périodiquement les reproducteurs. Et si on a un doute sur le fond génétique, on peut utiliser du génome scanning pour confirmer le fond génétique. Prochaine diapo. Ici, j'ai répertorié les erreurs qu'on rencontre le plus souvent au niveau de l'élevage à notre animalerie. Alors, malheureusement, il y a encore des problèmes de régie et d'inventaire, ce qui fait que des souris qui devraient être retirées de l'élevage sont gardées trop longtemps. Ces souris-là vont avoir de petites portées ou n'auront pas de portée. On a aussi le génotypage qui tarde trop, ce qui fait qu'on va établir les trios ou les couples tard et ces animaux-là vont avoir une baisse de leur performance reproductive et on va garder des souriceaux inutilement. L'autre problème qu'on observe, c'est que les problèmes de santé ne sont pas rapportés. Donc, le fait qu'on n'ait pas de génération, qu'on perd des portées, pas de gestation, pardon, qu'on perd des portées, qu'on a des petites portées ou de l'agressivité parmi les souris, on ne devrait pas considérer ça comme des problèmes normaux sans les investiguer. Et il peut être possible de corriger ces problèmes. Prochaine diapo. Il est très, très important d'avoir des bons dossiers. Donc, il est recommandé de faire un élevage à toutes les semaines. Oui, c'est beaucoup de travail, mais c'est payant de le faire. On devrait savoir le pourcentage de gestation qu'on a dans notre colonie, le nombre moyen de souriceaux sevrés par portée, le nombre de portées reproductives qu'une femelle peut avoir au cours de sa vie, la durée de reproduction des mâles et le numéro de génération de la colonie. Ça permet d'établir combien ça prend de paires ou de trios pour produire les animaux expérimentaux, à quel moment il est temps de remplacer les animaux reproducteurs et à quel moment il est temps de faire le rétrocroisement de la lignée sur le fond d'origine. Prochaine diapo. On a tendance à utiliser traditionnellement des trios pour élever nos souris, donc un mâle avec 2 femelles, mais cette stratégie d'élevage a été mise en doute. Il semblerait que les duos soient plus productifs et que les souriceaux issus de duos plutôt que de trios auraient moins de comportement anxieux. Je crois que ce qui est important, c'est de compiler nos stratégies, nos statistiques d'élevage et de choisir la meilleure stratégie. Pour les gens du centre de recherche, on vous encourage à utiliser l'Aptrax pour faire vos inventaires d'élevage parce qu'on peut en tirer des rapports et connaître nos statistiques. Prochaine diapo, s'il vous plaît. Ça peut être intéressant aussi de mieux planifier la production. Donc, première étape, il faut déterminer tous les génotypes dont on a besoin, autant des animaux expérimentaux que des animaux contrôle. Déterminer la meilleure stratégie pour obtenir tous ces génotypes-là. Et ensuite, utiliser des formules pour calculer la production. La formule que je préfère utiliser, ce sont les feuilles de calcul qui sont fournies par Jackson, mais il y en a d'autres aussi qu'on peut utiliser. Prochaine diapo. Alors, je termine en vous parlant de l'importance de la nomenclature. C'est très important d'utiliser la nomenclature appropriée, d'en connaître les règles et essayer de comprendre qu'est-ce que veut dire le nom d'une souris. Ici, j'ai mis le nom d'une souris transgénique et je vous ai mis en couleur les différents éléments de du nom de cette souris-là. Et c'est vraiment important d'utiliser aussi les bonnes abréviations et d'utiliser la bonne nomenclature au moment où on fait la publication des résultats. Christian vous a parlé du passeport souris. C'est aussi un document qui est très important, qui est promu depuis plusieurs années maintenant au sein de tous les gens qui utilisent des animaux modifiés par génie génétique. Ce document-là devrait comporter tous les éléments importants à la lignée, donc la nomenclature complète, le fond génétique, comment on va faire le génotypage, les besoins en régie, en protection du bien-être de ces animaux-là et aussi le phénotype. Alors, je vous remercie de votre attention et je vous souhaite une bonne fin de semaine.
Merci, Hélène. C'est des bons conseils pour tout le monde. Merci. Suivant, c'est Jean-François. Encore, j'essaie de faire le remote control.
Let me see if it works.
Parfait. Parfait.
Yes, it does. All right, so hello everyone. The topic of my part of the workshop will describe how new technologies such as CRISPR-Cas9 allow us to generate novel rodent models at a pace never seen before. My presentation will be divided in two sections. The first one will describe how these new technologies have revolutionized the way we create rodent models. And the second one will try to address one of the major drawbacks of these technologies, which is the possibility of undesired off-target mutations. I'll basically try to answer two questions. The first one is, should we be worried about off-target mutations? And the second one is, how can we mitigate their potential effect on the phenotype of the resulting phenotype of the newly developed animal model? But before going any further in my presentation, I would like to give you a brief crash course about how rodent models have been generated historically. That way you'll have a better understanding of how CRISPR-Cas9 is making our life easier nowadays. So, sorry, there's a lag. Okay, so in the literature, there's generally two types of animal models that are being described. So you have the first one being the random, well, the first one that I'm presenting would being the random integration transgenic animals, in which case a researcher would come to us with DNA, their transgene of interest that he would want us to insert in the genome. And so we would proceed with purifying the DNA, linearizing it, and then insert the transgene in the genome by pronuclear microinjection. In which case we would use one cell stage fertilized embryos, and we would proceed to inject the transgene into the male or female pronucleus of this one cell stage embryo. These, the microinjected embryos would then be implanted in pseudopregnant females, and the resulting pups would be characterized for absence or presence of the transgene. So this method has been shown to be relatively easy and rapid to generate novel animals. However, you have no control over the insertion site as well as the copy numbers inserted in the genome. Back in the old days, when researchers wanted to target a specific locus, they would rely on gene targeting in embryonic stem cells, in which case the construct used was much more sophisticated, as it would comprise extensive 5' and 3' homology arms, as well as in antibiotics resistance cassette. And so what we would do is we would electroporate this DNA construct in embryonic stem cells, perform selection with antibiotics, isolate positive clones, and then use conventional molecular biology techniques to characterize the insertion sites. And then the most interesting candidate would be injected into blastocyst, which is depicted here on the right side, which basically show targeted embryonic stem cells being injected close to the inner mast cells of blastocyst, which is a pre-implantation embryo at a later stage of development. So this method has been shown to be efficient at controlling where the construct would be inserted in the genome, as well as the copy number inserted in the genome, but it has also been shown to be highly tedious and labor intensive. And in both cases, the phenomenon, So in both cases, the phenomenon that allows us to generate new animal models rely on intrinsic mammalian DNA repair mechanisms that have one event in common, which is the presence of a double-strand DNA break. So when there's a double-strand DNA break in the genome, there's two pathways that can be used to resolve that break. There's either the non-homologous enjoining pathway, which is highly active. It's active in dividing and non-dividing cells, and from which the integration occurs the integration rate occurs frequently. It is the pathway that has been shown to be used in random integration transgenic animals, whereas on the other side, you have the homology-dependent repair pathway, which is a much more refined and specific pathway that's only active in dividing cells, and in fact, it's only active in a certain phase of the cell cycle, from which the targeting event has been shown to occur infrequently, and which is one of the reasons why when a researcher or a scientist wants to introduce their construct at a specific locus, they historically relied on embryonic stem cells. As we get millions of those, if your targeting events occur infrequently, you increase the likelihood that you'll get your construct properly integrated in the genome. But bottom line from this slide, what we have to remember is that scientists have known that if you're able to control where and when the double-strand DNA break will occur in the genome, then you'll have a control over your gene targeting project, basically. And so this is one of the reasons why scientists have been interested in control where and when the DNA break will occur in the genome. And historically, there's 4 different methods that have been described in the literature. 3 of them rely on protein-DNA interaction involving either meganucleases, transcription activator-like effector nucleases or TALEN, as well as zinc finger proteins. And all these 3 approaches have been shown to be somewhat efficient at performing their task. However, it has also been shown that they require extensive protein engineering, which is nothing in comparison to the simplicity of the CRISPR-Cas9 system. In this case, CRISPR stands for clustered regularly interspaced short palindromic repeats, which is a mouthful, and Cas9, which is the CRISPR-associated protein. And so this system came as some sort of a revolution in the transgenic field, as it relies on simple base pairing homology to confer the specificity of the cut site. And this is mainly due to the fact that this system only requires only 2 components, in which case it requires the Cas9 protein, which will induce the double-strand DNA break in the genome, and a single guide RNA or cRNA tracer RNA, depending on the formulation that you're using, that will confer the specificity to the system. And this is done due to the fact that the single guide RNA can be programmed or engineered to recognize a specific site in the genome, in which case it will recognize a site of a stretch of about 20 nucleotides. And it not only requires to recognize these 20 nucleotides to induce double-strand DNA break, but it also requires the presence of what we call a protospacer adjacent motif, or PAM site, just aside of the 20 nucleotide site, basically. And upon recognition of these 20 nucleotides along with the PAM site, the Cas9 protein would proceed to induce a double-strand DNA break 3 nucleotides upstream of the PAM site. And so knowing that, we can now realize how easy it is for us to design repair template that would allow to introduce a specific construct in the genome. And not only that, when we consider it, everywhere where there's an NGG sequence, there's a potential for guides to be designed that could be used in a gene editing or gene targeting project. And in fact, nowadays, this system has become so easy to use that we can apply it for various type of animal models that we need to generate. Within a spectrum of the easier project versus the more difficult one, any type of project that rely on the non-homologous and joining pathway tends to be easier to complete, whereas the one relying on the homology-dependent repair pathway still represent a bit of a challenge, but it is nothing in comparison to the work that it used to represent when we used to work with embryonic stem cells. And so in terms of easier project, any kind of project that will rely on or require inducing random mutation in the genome is relatively easy to perform. Also, what we call the classical deletion models, where an exon would be excised from the genome, is relatively easy to complete. Whereas the a little bit more difficult project, including a specific point mutation, can still be quite effectively completed. And in this case, we will rely on a repair template that contain the specific point mutation that would be introduced by a homology-dependent repair mechanism. And then finally, down the road of a little bit more difficult project, we have these somewhat large construct integration project, such as EGFP reporter or conditional alleles. But knowing that this system is very flexible as it allows us to generate various type of animal models, it is no surprise when you search for CRISPR mouse or CRISPR rat in the literature that there appears to be an exponential growth of publication related to these projects. And this is coming or starting from the point where people have realized that the CRISPR-Cas9 system was active or could be used in mammalian cells. And so obviously for any type of new technologies, there's always some concern about, undesired effect. And I guess one of the easiest drawbacks or comments that a reviewer could mention if you're trying to publish a paper about your new developed animal models would be, well, what about the off-targets? Did you look for off-targets in your animals before publishing the resulting phenotype? And so the next part of my talk would be try to address whether or not how we can control for potential off-target mutations. What is the prevalence of off-target mutation from a transgenic standpoint? And then finally, I'll conclude with my recommendation or suggestions when you get a newly developed animal model in your institution. But firstly, what are we talking about when we're talking about off-target mutations? So as I mentioned earlier, the specificity of the systems rely on the single guide RNA, which has a stretch of 20 nucleotide that could be engineered to recognize a specific sites. But it has been shown in the literature that some base positions or this system can also sometimes tolerate some mismatch within the 20 nucleotide sequence. This depends highly on the complexity of the sequence or the target sites that you're looking at. And so, in other words, you would get some events, very frequent event, where a double-strand DNA break will occur on target. And also, sometimes you may have undesired off-target cutting that could induce off-target mutations. And so one of the questions would be, well, how do we control for these potential undesired off-target mutation? One way to do that is by using prediction software, in which case we're using in silico prediction softwares. In our lab, we use 3 different available software. We use Chop, CRISPR, and Breaking-Cast for designing our projects. And these all rely on a similar approach to design the guides, as well as give prediction score for these guides. And I'm not going to go into details about describing how they work, but basically the bottom line, the way we normally do is you would input your DNA sequence of interest, and the software would proceed and scan its sequence and give you potential cutting sites or guide sites that are with prediction scores. And so the next slide is just to give you an example of the kind of results we would get from one of these software, in which case this is the result from CRISPR. And highlighted in the red rectangle box, you can see an example of these results. So on the left side, you have the guide identifier, and then the second column would give you the 20 nucleotide sequence along with the PAM site aside of it. And then the other columns that interest us when we're designing a new experiment are specificity score. So you have the MIT specificity score as well as the CFD spec scores. And basically what they tell you is, roughly what they tell you is how good your guide's going to be at recognizing its target sites versus potential of targets. And usually the higher the score, the better the guides would be at performing its task. You can also look for predicted efficiency score, which are somewhat reliable, but basically what they will tell you is how good your guide is going to be at inducing double-strand DNA break in the genome. And then the last two columns that usually really interest us are the number of potential of targets, depending on the number of mismatches. And then the last column, which to me is very interesting in general, is that you can look for off-target mutation, but predicted off-target mutation, and by selecting either the ones that are found in exon onlys or on the same chromosome as the one you're trying to target. And so I think in general, people who might be interested in potential of targets in exon only would be interested in making sure that there's no predicted of target mutation in another gene involved in their pathway of interest. And the reason why we might be looking for potential of targets on the same chromosome as the one you're trying to target is that you would want to avoid potential of targets that could be in linkage in your newly developed animal model. And so this is just, the next slide is just briefly to show you what these kind of results look like. So if you scroll over, you can get the, you can easily see the guide sequence, and then below it's the off-target sequence with the mismatch highlighted with the asterisk, as well as it gives you the chromosome coordinates. So it now becomes really easy to design PCR primer to amplify that region of interest and look to see whether or not there was any off-target mutation in your newly developed animal model. So now that we know how to sort of control for these potential off-target mutations by using in silico prediction software, the next question would be, well, how prevalent are they based on what is known in the literature? So I've looked, and I must say that there's not a big body of literature in terms of large-scale analysis, but I've been able to find 3 interesting publications that were published recently. 2 of them use whole genome sequencing for their analysis, and 1 of them use whole exome sequencing for their analysis. And so overall, basically what I've done collectively, it represents sequencing in 15 different targeted animals, so that animals that had Cas9-induced mutation in them. That represents 7 different guide RNA that were used, and they compare the sequencing data to a total of 6 control animals. And so bottom line, what comes out of these 3 publications is that they were able to find only 4 insertion deletions that were attributed to Cas9 off-target mutagenesis. And out of these 4 insertion deletions, all of them were predicted by in silico software, which means that in general, these perform their task very well. What's also interesting is that it has been shown that for one of these studies, they show that the insertion deletion that they found was not transmitted to the next progeny when they were selecting their targeted mutations, which means that we can breed out off-target mutation by performing simple backcrosses. And lastly, this particular paper showed that there was no statistical differences between Cas9-induced off-target mutations in comparison to normally occurring de novo mutations from one generation to another, which suggests that the rate of Cas9-induced mutation is relatively rare based on their analysis. In terms of large-scale studies, as I mentioned, so far, to my knowledge, there's no paper that have been published, but I was able to trace back an abstract that was presented by a group in Toronto, and I'm hoping, I think that they are planning on publishing a paper out of it. And so basically, this is an effort from the Knockout Mouse Phenotyping Project, COM2, along with the group in Toronto, in which they've performed whole genome sequencing on 51 Cas9-derived founder mice, representing 162 different guides. And they compared these 51 Cas9-derived founder mice to 25 inbred control mice. And so within the abstract, you get all the information about how they've performed their computational analysis. But bottom line, the conclusion from their presentation is that with appropriately designed Cas9 guide RNAs, off-target mutagenesis is rare. And most importantly, they show that genetic drift within a carefully maintained inbred line of mice results in thousands of genetic variants between individuals within that line. What that means, in other words, what that means is that the perfect or the appropriate control mice in your experimental procedures are control or litter mates. And so now that we sort of know that from the literature, it appears that off-target mutagenesis using CRISPR-Cas9 appears to be relatively low. But let's say that you're still concerned that there could be some potential off-target mutations in your newly developed animal models. The question is, well, what can you do to sort of try to detect them? And so there's two recommendations that I could make, if the slide could change. Yes. So how to detect the potential off-target mutations. So one way to do that could be to do targeted resequencing. So as I showed previously, the in silico prediction software are usually pretty good at giving you where the predicted off-targets could be found in the genome. So it's relatively quick and easy to perform. It's inexpensive, it requires PCR and then normal Sanger sequencing, but it depends on the in silico software prediction specificity. As I've shown, at least so far from what we know from the literature, they usually perform really well. But then the other approach that you could consider, which is more expensive, would be whole genome sequencing. However, it requires computer power, bioinformatic knowledge, and a proper analysis pipeline, but it would give you an unbiased results and a complete pictures of what you have in your hands, basically. Now what I want to do is just wrap up my presentation by giving you a few of my recommendations or suggestions. So the first one would be, depending on, usually if you get the animals from us, we're pretty good at sequencing verified founder animals. But if you get them from an outside distributor or company, make sure that they've sequence verified the founders. And if not, do it yourself. Then the other thing I would recommend is sequence verify the germline transmitted animals. And the reason why I'm mentioning this is that founder animals resulting from CRISPR-Cas9 tend to be mosaics, which means that they would have a mixture of different alleles. And so what you would see on a gel doesn't necessarily translate in sequence. So it's always better to sequence verify your germline transmitted animals. And lastly, in terms of recommendation, my rule of thumb would be backcross your animals before generating publishable data. I would say up to 5 generations, although some of my colleagues would say 2 to 3 generations is fine. If possible, work with strain originating from more than one founder. If possible, work with 2 independent strains generated using 2 different guides. You could test for predicted of target mutations, I think especially the one in linkage. And finally, if you have the resources and the budget, you could perform whole genome sequencing. Now I will leave the stage to Dr. Thierry Alquier.
Merci, Jean-François. Terry, I have passed the control.
It looks like it's working. Thank you. I'm just going to change, if I can.
Wonderful.
My pointer option. Oops.
Oops. All right. Let me start the share again. There you go.
Okay, thank you. Hi everyone. So I'm gonna give you some background on the Krelox system. Most of you, I think, are aware of the principles, and then I'm gonna guide you through some of the important aspects to consider when using this system and generating the animals, and also some recent issues that came out in the literature that I think we need to be aware of to basically better control experiments. So the Krelox system is based on a Kre recombinase. It's a DNA is basically an enzyme that was isolated in bacteriophages, the virus that infect bacteria, for its ability to recognize, bind LOXP sequences in the DNA. And so there are basically two scenarios. One in which the LOX sequence are either pointing to each other or in line with each other. And in that first case here, when Kre is recognizing the LOX sequence, it will invert the DNA sequence in between the LOX sequence. This is a principle that is really interesting and has been extensively used to overexpress genes in a Kre-dependent manner. It's been used in optogenetics and DREAD studies in neuroscience. And the other scenario, which is of interest to us here too, is when the flock sequence are basically in the same direction. Kre is going to recognize the LOX sequence and excise basically the gene or the exon in between these sequences. So when you generate, when you generate a LOX mouse, and you breed it with now a transgenic mouse expressing Kre under the control of a specific promoter. It could be a cell-specific promoter or tissue-specific promoter. You then have the ability to have basically tissue or cell-specific knockout. Basically, the Kre recombinase is going to be expressed only in the cells or tissue in which this promoter is active, and then recombining only the LOX gene or exon in your tissue of interest. And in that case, obviously, the knockout or the timing of the knockout is going to be dependent on the temporal expression pattern of the promoter that drives Kre expression. So now, as Jean-François mentioned, it's quite easy, I would say, to generate flocks animals. There are several companies that can do it for you, and there are also several consortium basically that generated a lot of flocks allele, including the International Knockout Mouse Consortium. And so when you ask a company or to generate a flux mouse, or you buy, let's say, the ES cells or the mouse from the IKMC, what do you get? So this is here an example in which the exon 2 of your gene of interest has been flanked by the LOX sequence, and your flux mouse is going to look like that. Basically, you're going to have, like, within the sequence of your genome of interest, obviously the flux sequence, as I mentioned, but you will still have some cassettes that are remaining or that are there that were used for the ES cells selection after the homologous recombination. And typically it's a LAGZ and antibiotic selection cassette, neomycin. And typically now, at least the new constructs, these cassettes are surrounded by other sequences that are called FRT that can be recognized by another DNA recombinase. And such you have two, and as such you have two basically options. Either you cross your Flox mouse directly with your tissue-specific Cre mouse, and in that case, Cre is doing its job. It's going to excise exon 2 within the lock sequence, but it's also going to excise Neo here because there is a Flox sequence in between LAC-Z and Neo. So you end up with a mouse without the exon 2, but you may still have one of the cassette remaining. The other option is to cross your Flox with a FLIPAS. It's available at Jackson, and it's going to be a two-step, basically. So you will have a first breed or cross with a FLIPAS mouse to remove this LAC-Z and Neo cassette, and then the resulting mouse, in which you have only the exon 2 with the Flox sequence, can be then crossed with your Cre-specific mouse to induce the knockout. So obviously there are consideration to take here about the genetic background of the FLIPAS and the Cre, and which background was used for the ESLs, and I'm going to come back to that after. So I won't go into the detail because there are obviously different possibilities, but as a rule of thumb, there are some, I would say, gold standard breeding scheme for the Krelox system. Once you have your Cre mouse, let's say from the Jackson lab, you got your Flox mouse generated or bought from or you obtain from a collaborator. The first step is obviously to breed the Cre with the Flox. And typically what is done is you're breeding hemizygous Cre carrier, meaning that there is only one allele that contained the Cre. The other allele is wild type. And you typically breed it with a mouse that is LOX positive and wild type on the other allele to get basically a Cre LOX plus offspring. And these mice are going to be used for the second round of breeding to obtain your experimental knockout in the step here. And you have there two options. Either you breed your Cre LOX plus with another LOX plus or with an homozygous LOX. And obviously, that's going to affect what kind of genotypes you're getting in the offspring. So in the first case, you're going to get, so I put here the stats, basically 1 out of 8 mice are going to be your knockout of interest. And then you're going to have a mix of genotypes. You're going to have heterozygous knockout. You're going to have Cre control. You're going to have some LOX heterozygous and some LOX homozygous and fully wild-type mice. In the second scenario, you're going to have an increased probability of getting knockouts, a lot of heterozygous mice and LOX control, but there is no Cre control or total wild-type or wild-type mice basically generated. So what I like about this scenario here is that you generate basically all your control litter mates. Obviously, it's difficult to have all the genotypes within 1 litter, but you use the control litter mates. And it also avoids genetic background drift if you're working with a mixed background. The cons are obviously, yes, you're not getting a lot of knockout mice, but I think that's the price to pay to basically generate your control in the best way. So some tips regarding the breeding scheme. We typically recommend to use the Cre carrier would be a male. The idea, the rationale is that most of the Cre mouse have a phenotype, and most of them have been studied in male, and we have no clue about potential phenotype emerging during the pregnancy in a Cre positive dam. So to avoid that, we use the male as a Cre carrier. You should not breed together Cre animals. The idea is to avoid homozygosy at the Cre locus, and I'm going to come back to that because Cre is potentially toxic. And finally, when you're following this breeding scheme, you don't want to use F2 mice to basically replenish your breeder. You should have separate colonies to generate your Cre breeder, your LOX breeder, and always use these initial animals to regenerate, as Hélène said, to regenerate your breeders. If you're using animals from the F2, in which you have a potential genetic drift, and you put back that as the breeder, then your colony basically in 6 months or a year is going to be different from the one you initially generated. So now I'd like to insist on one aspect that is really important. It's the genetic background. As you may know, there are dozens of different mouse strains. 129 FVB, DBA, Black Cis as the main one you've heard about. And these mice have major genotypic and phenotypic differences, whether we're talking metabolism, immune system, behavior, they really strong differences. And most of the Krelox animals you're going to generate, they're going to be potentially on a mixed background. Why? Because the LOX mice are typically generated using ESLs from 129 mice, or Black Cis N mice that you're going to breed with a Cree mouse from JAX, which is on a 6J background. So you end up with mixed background. And as such, you don't have control on the mix, basically, of the background. Is it's more 30% versus 70% on the 6J. As such, the phenotype of your experimental animal is going to be highly influenced by the genetic background. And one great example that was published not long ago is, if it wants to show, sorry, here we go. So this is a really nice paper in which they studied the impact of the genetic background on 2 different mutations, and I'm showing you here. It's a knockout for a calcium voltage gated channel, and they introduced the knockout basically in 30 different strains. And in that case, they just look at the locomotor activity induced by an injection of amphetamine. And the first striking results is that basically the effect of the methamphetamine on the knockout is totally different, I mean, not different, but at least dependent on the genetic background. You can see that the basal response is highly variable. And on top of that, in some animals are highly affected, such as this one, and some basically have strictly no difference at all in the phenotype. So that's a great example of how the background is going to basically either emphasize or enhance or silence the effect of the mutation on the phenotype. And, oops, sorry. And this problem is even more important when you consider the Black Cis strain. You know, when you read a paper, typically you look at the method and you're going to see, oh, we used, or the mice are on the Black Cis background. But here, the idea, I mean, what I want to show you is how many substrains of Black Cis exist actually around the world. So the first original Black Cis strain emerged was generated or actually came from the Jackson and in the 50s here. And this is what is called the Black 6J. But then this mouse strain was also acquired by Charles River. And in their own farm, they established a colony, which is now the Black Cis N. And these mice basically went all over around the world, in Japan, in Asia, in Europe. And so you have different providers. And as such, you end up with like many 6N substrains and many 6J substrains. And in this study, what they looked at is at basically the specific nucleotide polymorphism, SNPs, in the Black Cis J compared to other strains. And you can see that there are a lot of polymorphism differences between the substrains. And here are represented only the polymorphism. There are many reports in the literature showing that there are complete deletions, gene deletions. There are also retrotransposon insertion, one affecting particularly the raptor, the gene coding for raptor. And there are also copy number variation for several genes, including the insulin degrading enzyme and some binding protein for FGF. And I want to show you one example on how the background, the different substrains of the Black Cis is going to affect the phenotype. It's well known in the literature that there are mutations in the 6N and the 6J background. And something that is important, at least for what we are studying in metabolism and insulin secretion and beta cells, is the Nnt mutation. And this is a mutation that was identified in the black 6J background. In the gene, it's a deletion in the gene coding for the nicotinamide nucleotide transhydrogenase, known as the NNT gene. And this enzyme is involved in the transfer of the hydrogen between the NADH and NADP in the inner mitochondrial membrane. And as such, when you don't have the enzyme, there is deletion, you have a reduced NADPH content and altered mitochondrial function. And what we've done, and we were not the only one to show that, just looking at basically how much insulin this 6J versus 6N are secreting in response to glucose. This is one study we've done with the phenotyping core facility with Grace Ferguson and Melanie Atelier, in which we just compared insulin secretion during basically provoked hyperglycemia. And as you can see here, the black 6N have a beautiful insulin secretion, first phase, second phase. The black 6J, it's absolutely flat. They do not secrete insulin. And so you can imagine when you're studying the mechanisms involved in type 2 diabetes, for instance, that will have a major consequence on how you interpret your data. And the group of Mark Prenke even showed, like in the same line, that mice that are heterozygous for the mutation, that are NG, they have one wild type allele and the Nnt mutation, they still have a defect in insulin secretion. And so that's just one example looking at one function. But there are others that are described in the literature. So what are the solutions? How to deal when you have a mixed background? The first one is to backcross. So there are two ways to do it. Either you do a regular breeding by breeding for several rounds with basically 6J or 6N mice, or you can speed it up by using in vitro fecundation. But you have to choose, obviously, on which background you want to backcross. If your Cree is from Jackson, the obvious choice would be to backcross your flocks on the black 6J, and then you have a pure background in your knockout. You could decide the other way. You could change the background of your Cree and decide to backcross it on the 6N. But you have to know basically that every strain or substrain you're going to use is going to come with a set of mutations that may eventually affect what you're looking at. For instance, the 6N have a mutation in the gene coding for CRB1 that make them more susceptible to retinopathy. So the best is to do your homework, basically, get information and choose the background wisely. And the second option is there is no problem in studying and keeping animal on mixed background. What you want to do is to limit the genetic drift and always use control intermates. And eventually you can genotype for the mutation that are known or potentially known to affect what you're looking at. And you can select or remove the animals that have this specific mutation. And what is really important is, and as Christian said and Hélène, is to provide a detailed and complete information on the background strain and the breeding practice and the control groups. As it was recently discussed in the literature, there is, when you look at the methods in the paper, it's really concerning because basically it's only 60% of, or 40%, sorry, of the methods that clearly identify the background, the specific background of the model in the scientific literature. I'm stuck, Christine. I don't know why. Oh, okay. So I want to give you quickly some input on the most appropriate control for the Krelox system. I think something that I strongly believe is to include Cree controls. Why? Because typically the Cree mice have been generated by the classical transgenesis, and it's inserted randomly in the genome with a number of copies that is unknown. And most Cree strains, because of that, have a phenotype. The second point is that the genome contains what we call degenerate LOXP sites that are recognized and recombined by CRE, leading to off-target recombination of genes in a tissue-specific manner. So there was one paper that looked at that. They identified more than 600 LOXP sites, more than 200 overlapped with annotated genes, and 55 of these genes were expressed in the cardiac muscle. And that was a mouse, the cardiac-specific CRE mouse. And they found that the expression of 20, basically 1/4 of these 20-something genes were disrupted in the cardiac CRE-expressing mouse. So that gives you a good example of how important it is to include CRE control. What about the LOXP control? So sometimes the LOXP sites are not silent, and they may affect the expression of the endogenous genes. That's something important to control, and to potentially include your LOXP as control if the expression of the endogenous gene is affected. The heterozygous knockout mice, I think they're also important because they help you assess a potential gene dosage relationship with the phenotype. And they're also more representative of pathological conditions in which gene expression is reduced. You know, in disease, we don't have like, except monogenic disease, we don't have like a complete ablation of the gene expression. And then finally, the wild type, you can basically compare them to your flocks and include them as a whole group of control. So I won't go into the detail of every issue that was reported regarding the Krelox system. There are two examples I want to talk about. One is the expression of the human growth hormone genes from the CRE transgene, and the other one is the ectopic CRE expression during gametogenesis. But there is a list here of several issues that have been reported regarding the toxicity, the leakiness of the CRE expression. And there are here some excellent reviews that I would encourage you to read about these issues that were recently discussed in the literature. Or something I know it's already 1:15, we are beyond, but this is really something, the ectopic CRE expression, I really want to address because this is a big issue, I think, in the field that is totally silent. So here is your, let's say, your neuron-specific CRE mouse and your breeder to generate your knockout animals. It's a CRE LOX heterozygous mouse. Typically, what we do to genotype these animals, we have a typical genotyping PCR strategy with two bands. So basically, the difference you're going to see on your gel when you genotype those guys is the LOX sequence. So you're going to have one primer on the exon that is supposed to be deleted, one primer in the intron, and then you're going to amplify this band. And with the flock sequence, the signal is going to be longer. But in that case, if there is a recombination, the PCR won't amplify anything because the exon is gone. Okay, something to keep in mind. So the ectopic CRE expression during gametogenesis is basically a random phenomenon that happens depending on the strain and the sex, in which the CRE is going to be expressed during the spermatozoid or the oocyte production. We don't know why, but it's going to happen, and CRE is expressed, is going to do its job. So you're going to end up with a spermatozoid and oocyte in which the exon is already gone. Okay. And when you breed your Krelox with your typical LOX heterozygous to generate your knockout, you're going to end up with mice that are CRE positive. They may inherit from one allele in which there is already recombination not occur with this allele, wild type. And if you genotype these animals using this two-band PCR protocol, you're going to see what? Your primer are going to tell you the animal is CRE, okay, and it won't see the recombination, okay? It's going to see only the wild type allele. So you're going to conclude, oh, that's a CRE control. But in the end, it's a whole body heterozygous knockout mice. You can see how that can impact your research. And if that allele was recombined with, associated with this one, your genotype is going to tell you again, doesn't see anything here. It's going to see like a flux allele, the CRE, and it's going to say it's a CRE LOX. It's a knockout, but again, it's also a whole body heterozygous knockout. So all that to say that the ectopic recombination is really an issue, and the best way to control for it is to design, when possible, obviously, a free band PCR that will give you a signal even when the recombination occurred. We faced this issue in our group. For one CRE mouse, I can tell you it's painful. So if you have ways to design the PCR that way, to detect the recombination once you genotype the animal, that's definitely the way to go. And I highly encourage you to read this paper in which they made a list, like dozens of CRE mouse in which the germline recombination was quantified. And you can see in some strains, such as here, the synapsin CRE, which is a typical neuronal CRE mouse, it's 60% of the offspring that are going to show the delta allele, this ectopic recombination. So this is really critical. And Jackson is even now reporting it on the website. And they are, to decrease that frequency, they suggest to put actually the CRE transgene into the female instead of the male. And there was also, again, a new case of germline recombination described in a CRE mouse used in immunology, the FOXP3 CRE mouse. So this is really like something that is overlooked. The final thing, what I want to conclude on is the alternative to avoid some of these issues. And the first one is the tamoxifen inducible system. Yeah. Yeah, I know. I won't go into the detail, but it's a way to basically trap CRE into the cytosol. And once it binds tamoxifen, it's going to migrate to the nucleus and then do its job. Tamoxifen is an estrogen receptor agonist. So you can imagine it has side effects. So it's always better to use some washout period after the treatment of the animals to make sure there is no issue with that. Often there is a low recombination efficiency, and there is also some spontaneous activity without tamoxifen. Another system that is used also is the TET-on or TET-off system, tetracycline inducible or repressed. In that case, you would need, it's a triple transgenic mouse. The specificity might be increased because you have two promoters basically, but there is the group of Jen Estal at IRCM is now showing that the transactivator expression has also deleterious effects and side effects. And there is the next generation of CRE that is coming, the photoactivable and the self-cleaved inducible CRE-ER. And I will finish on that issue, the growth hormone mini gene. Most of the transgene, the CRE transgene, is followed basically by the human growth hormone mini gene to enhance CRE transcription. The only issue is that in some strain, actually the gene is coding for growth hormone. And it has been shown that it acts biologically, specifically in beta cell. It's going to affect beta cell mass, function, proliferation, and many other things. It's been reported in other non-beta cell strain. It's a huge concern with the nesting CRE mouse, in which the phenotype of the CRE mouse is dependent on the human growth hormone. And I highly encourage you to read this review, in which there is the list of 300 CRE strains that contain the human mini gene, and then you can check whether your favorite line has it or not. So in general, for the guidelines for the Krelox system, the first thing is to seek information, get advice. We have great resources within the institution, the CIPA, the Transgénèse core. Use CRE homozygous breeders, check the status of the mini gene for the growth hormone. Optimize the genotyping PCR primers, use reporter strain to assess the CRE specificity, always include CRE littermates, and always use control littermates, and consider alternatives, obviously, and other strategies, and report. Always report what you have done. Thank you. I'm sorry for going over.
Merci, Thierry. Lots of good advice in there, and lots of good strategies, good practices of how to manage your Krelox. So I'm just going to wrap it up with from the 4 talks with a couple of really easy to remember take-home messages. The first one is one that I've, for those of you that know me, I've said it over and over. CIPA and the veterinarian team are your friends. They are there to help you before, during, and after your project. Make sure that you consult with them. They have a lot of information that can help you reach your research goals faster. The second message is one that comes out both in Helene's talk as well as in Thierry's talk, is that words matter. So making sure that you use the appropriate nomenclature for your mice is absolutely critical. That you use good breeding strategies in order to get the mouse lines that you want and to maintain the lines for the future. The third is also to highlight that the tips, tricks, and tech that they exist here. The CRCHUM, actually the transgenic platform under the guidance of Jean-François, is very, very savvy and up to date on all of the methods that are used. And in fact, Jean-François is actively innovating new methodologies and to make animal modelings faster. And he can provide turnkey solutions for you. Also, if you have plans to use Krelox strategies, Thierry is an infinite resource, as you just witnessed, and all of the things related to using those models. The fourth is know your limits. All tools have limits, all models have limits, and no model is ever perfect, but it's really important that you know what they are, that you go in eyes wide open, and you recognize those and how those limits might impact the interpretation of your experiments. And take-home message number 5 is be in the know. It's the responsibility of the researcher to know the details of the animal model that they're working with, and most importantly, to report it accurately. We are in a crisis of reproducibility in certain sectors of our research community. It is really incumbent on each of us to make sure that we report things fully in the literature. And the last message I have for you is all of these new technologies and new skills that have developed over the years, is that with great power comes great responsibility. And so the more we can manipulate the genome of our animal models, the more we must understand what we're doing and report that appropriately. So with that, I'm going to see if there are any questions sitting here in the Q&A. There have been several requests for whether or not the slides will be available. The answer is absolutely. The presentation was recorded, and the slides and the presentation recording will be available on the Transgenic platform website on Monday. Are there any specific questions for our panelists? The panelists, if you can turn on your cameras, please. There is one question from Eric asking if there is any advice on RMCE using CRE in a single or double lock site. Thierry, maybe?
Yeah, the RMCE is really a better way to generate the new CRE or the new generation of CRE models. It's basically a way to do a knocking. It's a targeted insertion of the CRE transgene. It could be knocking basically or replace. It's been done, for instance, I think about this example in beta cell research. You replace one of the insulin gene by CRE using targeted insertion. That way the CRE is expressed under the expression of the endogenous promoter of the insulin gene. For the single and double lock sites, I didn't go there, but we talked about it with Jean-François. One of the risks with double locks sites, I'm not sure actually, Eric, is what you're referring to. You're talking about a double knockout? Because that could be an issue when you try to have two genes knockout, two flux gene knockout, is that the CRE, if there is a proximity between the flux sequence, the CRE might actually take a big chunk of DNA in between the locks sequence. No, I can't comment on that. I don't know. Locks 71 and 66.
Okay.
Maybe Jean-François knows.
Locks 71 and 66. Those are mutant LuxP sites. I don't really know what's the question. Do you have the entire question, or?
He's asking
Okay.
for advice on RMCE.
Yeah. So if you're going for a double conditional knockout, so if you're trying to inactivate two different genes, so let's say gene A and gene B, and they're flanked by LuxP sites. So one, and this is to remember. So in Thierry's diagram, you have to remember that when your gene or your exon gets excised, there's always a LuxP site that remain in the genome. And so if you have exon A and exon B being removed, there's always the possibility that there could be a transchromosomal location between the remaining LuxP sites in the genome. And so what Eric is referring to in terms of LuxP2272, this is a mutant LuxP site that will recombine between two LUX2272, but will not recombine with the normal, the endogenous sequence, not the endogenous, but the wild type sequence of the LUXB site. So in my opinion, the recommendation is if you want to conditionally inactivate two different genes, you would use one animal model with gene A flanked by wild type LUXB sites, and one animal model with gene B flanked by LUXP2272 LUXB site. Don't know if it makes sense what I just said, but
if not, excellent. Eric seems satisfied, and otherwise he knows how to find you, that you guys can continue that conversation.
Sure.
Are there any other questions? We still have 50 people with us online. It's highlighted here, I see in the chat, to make sure that people are sharing their mice. It's important that we share it. There is another question from Yukihiro. Says, thanks for the great talks. Can we use CRISPR-Cas9 technology to create a knock-in mouse derived from a human mutation in Canada?
So I guess the question is, can you reproduce the human mutation in the mouse genome?
I believe that would be correct, yes.
So yes, we can do it as long as let's say if it's an amino acid that you're trying to target, as long as the amino acid is conserved, then most likely yes.
Excellent, thank you. An additional comment, Jean-François? Okay, are there any other questions? Yeah, I thought you were going to say something else and apologize. Was there any other questions from the audience? So not seeing any. I think I'd take this opportunity to personally thank all of you for preparing really, really thorough and very helpful presentations. Christian and Hélène, you guys are an infinite resource that I think the community needs to know about more. And so I really appreciate you guys putting the time in for this and sharing this. Jean-François, you've got a great expertise in the platform and have taken it to new levels, and I appreciate it very much working with you. And Thierry, a big thank you to you for really spearheading this and using this platform as an opportunity to open this conversation about how to improve reproducibility in research and how to use animal models effectively, and really putting on display the level of expertise that already exists at the CRCHUM. So thank you very, very much. And a special shout out to Mireille. Thank you, Mireille, for doing the behind the scenes organization for us. Very much appreciated. Très apprécié. I think that's all for us this afternoon.
Thank you all for listening, and thank you, Jean-François, Hélène, and Christine, because it was a great venture.
Wonderful. It's been a pleasure to be involved. Thank you so much for including me. All right, thank you, everybody. We'll sign off for now. We wish you a wonderful afternoon.